For our third speaker, he is an environmental archaeologist and paleontologist currently studying the effects of climate and anthropization on biogeographical histories, genotypes and phenotypes of various species of small mammals in Europe and Southeast Asia. He's also dedicated to the multi-proxy reconstruction of past environments along the Quaternary to study interactions between environmental change and human development. His other research interests also includes taxonomy, evolution, palaeobiology, biochronology, taphonomy, palaeoecology, and biogeography of small mammals from the Pleistocene and Holocene. And okay, let us all give a warm welcome to Dr. Juan Rofes.
Thank you, Ellie, for that presentation. First of all, thank you very much for inviting me. Maraming salamat. Magandang hapon, everyone. So thank you for giving me the chance to contribute at least my grain of sand to the celebrations of ASP 27th anniversary. And today I'm going to speak about new methods and technologies in zooarchaeology. So, okay. So up to now, you have these two visions. So digitizing, about possibilities of digitizing in archaeology. I'm going to talk from the point of view as zooarchaeologist, most of all, and I would like to talk about two groundbreaking methods using technologies that are really hallmark now in zooarchaeology. That those are stable-isotopes and archaeogenetics. Right? I'm not being a specialist on those. I will touch them at the end of my presentation, but I will start with technology that are more simple, maybe more everyday technology and not that fancy, but technology that have helped me during my career. Okay, so that's why I call it a personal account. So, and I want to start with Patapuerca, right? So this is a site that many of you have heard from me or you already know. It's a place there in Spain, and it's a key site for understanding human evolution in Europe in particular, and in the world. So this Sierra de Patapuerca is actually more than a highland. It's a hill that at the beginning of the 20th century was cut. I am trying to move there. So it was cut by a railway trench, and that railway trench accidentally revealed this amazing stratigraphy, these amazing infills of paleo caves, like this one, Grandolina. And not only this one, but also here, this other cave, Galeria, Cima del Elefante. Those are the exposed ones, but there are also active caves, like Cima de los Huesos. So in Grandolina, it was found in the 90s, mid-90s, the remains in the level 6, Trato Aurora. It was found the remains of Homo antecessor, right? More than 100 now, maybe more than 200 nowadays, because the excavations continue. I was part of the field work of Patapuerca during 18 years, but they continue digging there more and more. So Homo antecessor was found there in level 6. In Galeria, there's just few human remains, Homo heidelbergensis, but it's mostly a natural trap. You see the animals and the humans were falling there. And Cima del Elefante, this site in the lower layers, have the remains of the oldest Europeans, oldest direct evidence in the form of this mandible, 1.2 million years ago. It was deposited, and this year, where one of our students, Patricia Cabrera, was there this year, she was lucky because it was found remains of the face of an individual, maybe connected, maybe not, with this mandible, because the face is 1.4 million years old. Encima de los huesos, we have this fantastic accumulation of almost 30 individuals of Homo heidelbergensis, pre-Neanderthals, with skeleton skulls as complete as this, and the skeletons almost complete. Is the archaeological sites with the highest accumulation of human remains for the lower Pleistocene. But my side of knowledge in Patapuerca, my interest was mainly in, as you know, small vertebrates. This is like an accumulation of small vertebrates from Patapuerca, and they are recovered every year. So we have more than 7 sites being excavated at the same time. So we need a really intensive way of wet sieving the material. We wet sieve more than 25 tons of sediment per year. Okay, so we have this scaffolding structure there with regulable hoses and this stack of 3 sieves. Here I am presenting you a synthesis or an abstract of the process, right? So the sediment are put to deflocculate in especially the hard clays for them to soften in buckets with water. They are powered in the stack of sieves, 1 centimeter mesh light, 2 millimeters, 0.5 millimeters, so nothing will be lost. Then the sediment of these 2 sieves are put to dry, the coarse concentrates and the finer ones. They are put once dried into plastic bags and they are taken to our labs. But in the field, we have a field laboratory. We start sorting the material, so removing what is bone from what is small gravel, rocks. This is a concentrate of those bones already prepared for study and examples. So here, this is a mustelid, and this one is what we call a giant shrew. So you could say, why I'm calling this a giant shrew if it can stand in a finger? So because we calculated the mass of these animals in around 60 to 70 grams, when the biggest shrews nowadays don't reach 30 grams. So they were really big. And then we studied them in our labs. We use this microscope with a camera attached and a software that allow us to visualize, to view, and take pictures of them, take measurements. But in order to get a better understanding and to access even minimal details of these small creatures, we use the well-known technique called SEM, scanning electron microscope, like this picture. This is a maxilla, SEM maxilla of this same animal here. And here I will show you an SEM, as we say in Espanol, of Dolinosaurus glyphodon. This is a giant shrew that we described from the side of Grandolina. So what in a name? Because we describe it in this publication, this new genus and species of giant shrew. Sorex means shrew, so shrew from the site of Dolina. Glyphodon in Greek will mean teeth with a groove. Why? Because here there's a groove in the incisors, in the lower incisors. And we hypothesize that this groove in the lower incisor serve to deliver venom, salivary venom from the salivary glands, okay, venomous saliva that was delivered to the bodies of the prey, to not to kill them, but to keep them comatose and hoard them, in their little caves, underground caves. So we think that these extinct giant shrews had a similar behavior than the now existing short-tailed shrews from North America, right? And that the venomous injection apparatus delivery apparatus was similar to a endangered species from Cuba and Haiti called Solenodon or Almiki, local, because they also have incisors with a very deep grooves. Okay, so here with SEM, pictures, we can even access to see the groove in all its glory. This is this part, the point. And here we can also see from Dolinosaurus glyphodon the various toed mandibles, the symphysis of the mandible attaching the two hemimandibles, probably lies full of connective tissue, and reinforcing the various toed mandibles of these small animals for a more powerful gap. This was also published in 2007. And also through SEM, we could access this wonderful evidence of depressions in the proximal part here of a mole humerus, most probably caused by the bite of Benemendia ficides, one of the giant venomous shrews. Okay, why we think this was caused by a shrew and not by any other carnivore? Why? Because this was, thanks to these images, it could have been seen that the two holes are very, very alike, so same cause, and measuring the distance between them is exactly the same distance between the two upper incisors of a giant shrew, Benemendia ficides in this case. Measuring any other carnivore present in the side, no, the measurements do not correspond. So we adventured and we published this also as a very exceptional evidence of a bite, of the hunting of a mole, of the hunting of a venomous shrew on a mole. Here we can see it is even inside the two holes. But then we come back to our characters. All the characters could be very properly seen in these SEM pictures. And because we are able to describe so in detail, we can proceed with another kind of analysis. It's a phylogenetic analysis, but based on 48 homologous morphological characters. This is, we have to distinguish this from the molecular analysis. You can also perform this kind of analysis, but with base pairs, right? So with the material from the genome, we will do this kind of analysis. But in this case, instead of base pairs, we will use morphological characters. We will arrange the 48 morphological characters by states of character. So for example, teeth, pigmented or unpigmented, 0, 1, we assign a number, right? In this case, number of antemolars, 5, 4, 3, 2, 0, 1, 2, 3. Premolar 4, basin, posterolingual basin, deep or shallow, and so on. Then all these zeros, 1, 2, and sometimes 3, we introduce it in a matrix like this. So we can compare our specimen that we would like to understand the phylogenetic relationships with so many other shrews, extinct and extant, that we have the same record, the same homologous morphological characters. So a program called PAU, phylogenetic analysis using parsimony, will allow us, in those days, this analysis required overnight processing. Nowadays, I think it's much quicker. I have not done this since 10 years ago, this kind of phylogenetic analysis. So you will get a phylogenetic tree, right? And here is our Dolinosaurus glyphodon that have a common ancestor with a shrew, Tyrannosaurus leaf from China, and not with other giant shrews, not directly with other giant shrews from Patapuerca itself. Okay, so it seems to be connected to shrews there in Asia, primarily. And this, so the whole three have a low consistency index, so more relations needs to be demonstrated. But for this specific part, we did a reanalysis of just this part, and we got a high consistency index. This is the statistical support, more close to 1. So this we can trust this. Okay. So then I will show you the equipment and software that we use. This is the scanning electron microscope machine, how it looks like, and phylogenetic analysis using parsimony software. That you, well, this is an paid package. You have to pay for it. But in this site, in this online site, you can get a beta version for, I think it's free for 1 month, and then you have to pay for it. And now I will move to my postdoctoral experience in Paris, in France, specifically in the Museum of Natural History, Paris, where I got a Marie Curie scholarship to work on something called historical biogeography. So we wanted to understand how several species, groups of small mammals repopulate Northern Europe after a glacial episode. Okay, during glacial periods, animals inhabiting, in this case in Europe, Northern Hemisphere, inhabiting northern part of the continent, because of the advance of the glaciers, they will be pushed southwards. In this case, for this animal, Crocidura suaveolens, its most likely refuge was the Iberian Peninsula. This in green is the distribution nowadays, and you can see how the distribution nowadays gives us already data of what was its refuge during the glacial age. This is the distribution now during the Holocene optimum climaticum. And you see the distribution continental, it reaches up to there, but it's also present in these islands. Even in the continent, it is not nowadays. So one note to say that these islands that could be seen here better get separated from the continent around 10,000 years ago. From a process 10,000 to 2,000, they got fully separated. So the glaciers retreated and the sea levels rise. So they made it possible that those islands become isolated, and become islands, properly said. So we studied all this territory. We put in our samples to understand the biogeography of these animals, modern samples from the continent and from islands, Atlantic and Mediterranean, and archaeological samples. And because we want to understand, so I will go quickly back. So we want to understand how was the recolonization process before LGM? When and how did they get to the islands? And if humans have something to do with the arrival to the islands, or it was just caused by natural process. So we put together these samples, right? Archaeological and modern. In the case of the archaeological ones, some archaeologists have already published the radiocarbon dates to understand the chronology. But in these three cases, there were none radiocarbon dates. So then what we did was pair with another project that was implementing in Paris the installation and use of this machine called an AMS, accelerator mass spectrometer, Mikada's radiocarbon dating. Okay? And with this technique, just one mandible of a shrew could already be radiocarbon dated. In the past, we need a minimum of 60 grams of bone. That means, in terms of small vertebrates, it means a handful of bones, right? So destruction of all of them, and as they are so small, there could be percolation between different layers. So we can be inputting in the machine mixed material from different layers without knowing for sure. So it's much safer as we can date just one element that is safely placed in a layer. And we can even taste with this technology, ATR, FTR spectrometer, we can taste just 1 milligram of bone, would tell us if there is collagen enough for the radiocarbon date to be made. If it's negative, we will not destroy the mandible. If it's positive, we will be destroying just one mandible out of hundreds, probably, right? And we will be getting our radiocarbon dates, as we did. Then for the proper morphological analysis, we use a technique that, by contrast to morphoscopic morphometrics that you use just linear measurements, we use geometric morphometrics already introduced by Kim in the previous talk. So in this case, it's just a simple 2D geometric morphometrics analysis. Okay, we put landmarks, as Kim was explaining, and we introduce these landmarks, homologous point in all the pictures that we take of different mandibles, archaeological and recent, 9 landmarks in this case, we overimpose them with a software and we get consensus images. Here is the consensus for continent in blue and insular, in the islands, in red. And how much the different populations diverge, separate from the consensus is what we get here in terms of size and shape, because this technique allows us to study size and shape separately. So for the size, what did we found? We found that all the continental samples, so the continental specimens, were smaller than the insular ones, Atlantic islands, Mediterranean islands. Why? Because all of them were affected by what we call insular syndrome. Small animals, vertebrates, especially mammals, tend to increase in size in islands because they are released from their natural predators and their natural competitors, and they become bigger. And we demonstrated that in all these islands, the insular syndrome was already ongoing. And as for shape, we obtained these 3 well-arranged biogeographical groups: Atlantic islands, Mediterranean islands, and continent. Okay, so most of these distances are so long because these samples are modern or semi-modern, while the island of Molen samples came from an archaeological site, Neolithic Bronze Age boundary, and still the shape is closer to the mother population in the continent, while these others have more time to diverge until nowadays. Okay, but another anomalous thing, we find that these islands, Erik and Sark, had a shape similar to continent, right? They naturally cluster with a continental group. What was happening? So our interpretation is that the shrews regained territory during the recolonization process. They reached there, and then it came the rise of the seawater that isolated islands. So the shrews were naturally isolated there, and they diverged in shape up to here, right? While these other populations were more probably introduced recently by humans, most probably by Romans, just 2K years ago, okay? Because there are testimonies that the Romans arrived to these islands by that age, and probably accidentally they brought the shrews in their boats. Okay, also we found that because Molen is already, the specimens are already big, the changes in size are relatively quickly occurring, while the changes in shape, they take much longer time to take place. Okay, but also we wanted to understand what specific ecogeographical factors were affecting these populations for this change in size and shape. So what we did was collect the most popular in the bibliography ecogeographical factors that can affect, that can reshape the mandibles. Those are the area of the islands, distance in kilometers to the mainland, chronology of separation, latitude, longitude, altitude, temperature, precipitation, competitors. And so what we did was a linear regression analysis, and we get these results. So those marked in red and blue are the ones that have more statistical significance, meaning that, for example, the distance to the continent, chronology, latitude, and those have a more active role in shaping the mandibles than the area of the islands, for example. Most of them in the case of shape, and just 4 of them in the case of size. Okay, for the Mediterranean, we didn't have samples enough for them to produce satisfactory results. So, okay, regarding radiocarbon dating, the other side of the project, so the tiny size of small remains favors percolations into underlying layers along stratigraphic sequence. This is a well-known phenomenon in archaeological deposits. This new technique, Micadas, offers a straightforward possibility of detecting intrusive episodes through the absolute dating of minute amounts of bone down to 3 millimeters, meaning that isolated elements are enough to obtain reliable radiocarbon dates if collagen is moderately to well preserved. And we applied this in the site of Grand Rivoire, Eastern France. So the archaeologists already have a lot of radiocarbon dates, charcoal, seeds, large mammals. And what we did was contribute and input our radiocarbon dates from small mammals, from shrews, and compare what they already have. And it happened that all our data was in agreement, in perfect agreement, what they already had. That means that all studies regarding small vertebrates from these layers are to be trusted for biochronology, biogeography, and environmental reconstruction. Okay, but that was not the case in the site of Peyraset in the Western France. In this case, for the units 5434, there was a coincidence. So in blue, our dates were with shrews, and then they dated rodents and they dated large mammals like deer. There was a coincidence from our dates. But in unit 2, it happened that the radiocarbon date for deer was very different from our dated shrews. What does it mean? That means that we cannot trust anything coming from this layer because this could be contaminations, percolations. So any reconstructions, any biochronology done with this layer, from these layers, we can trust them, not this one. And it's important to know that because otherwise we can get to misleading results. Okay, all this data was published in several papers. And then, well, this is the equipment and software I use for the geometric morphometrics TPS, DIG and TPS RELW. The both softwares are free and you can access them in the online. For statistical analysis, size and shape, I use the R language, also accessible online, and Morpho-J, accessible too. And this is the AMS Micados radiocarbon data. Then let's jump to Southeast Asia, to the Philippines, where I have been the last 4 years. So I'm currently the head of the Zooarchaeology lab, as you all know. And this is our lab, some of our researchers doing studying of small vertebrates and large vertebrates, our collection of shells also. And I was totally targeted since the beginning to get an equipment like the ones I used to have in Europe here in the Philippines. So with the help of this project, Biodiversity, Evolution, and Palaeoecology, late Middle Pleistocene to Middle Holocene small mammals in Iran, funded already by Filipino funds, ORG, Outright Project, OBCRD, we managed to buy this equipment that cost more than around half a million pesos. So a microscope with a camera attached and a software that allow us to take this fantastic pictures. So the samples are there, right? And we can also take measurements. The program allows to directly export the measurements to Excel, plus many other features. And here I'm working in this case with materials from Callao Cave. So getting microphotographs and measurements from small vertebrates from Callao Cave, Cagayan Valley. Then this is an example of a plate that it's included in my upcoming paper for Middle East, Tangishikan, Zagros in Iran. And as you see, after playing with the remains with Adobe Illustrator, we can get images that are ready to publish, okay? Scale all of them. That's what we will do in due time with the material from Callao Cave, Luzon. These are photographs of insectivorous bat mandibles, a rodent molar, and a shrew molar, all of them from Callao Cave, currently under study. And then we can also use this equipment not only for small vertebrates, but also for details of large mammals in this case. This is material that I assigned to my students during my 207 class, from Musaya Cave, Biagnabato, Bulacan. Okay, all of this Callao and this material kindly given to us by Mandy. So we have here deer molar, occlusal and buccal views of a deer molar, pig molar, occlusal view and details of a tiny portion chipped off. We can get this degree of detail. This is a conventional photograph and a deer tibia distal fragment. Okay, we can appreciate the cut marks. mostly sure they were made by lithic tools, because we can appreciate the detail and the opening, right? The V shape of the marks. And in this case, it's we can also see this incision, this puncture probably made by a bite, okay, in this distal fragment of beard. Okay, so in the next one, I also opened the equipment and the lab to students doing analyzing other materials. In this case, you know, JJ Tablan was doing research on the sherds, the ceramic sherds coming from the Holocene layers of Callao Cave. So we have examples here. You see the size. This is 10 millimeters, very, very small sherds, plain earthenware sherds with incised box square marks. He could even take measurements also for these earthenware sherds with incised marks and measurements. And recently, Patricia Panganiban was taking pictures of metals, right? Metals and slags. Here we see oxidized surface metal implement, broken metal implement exposing iron oxide, right? And a detail of this part, detail of the previous show, sand particles, heterogeneous surface of metal slag, right? Metal slag with ferrous iron corrosion from leftover metal. And here a detail of the previous showing flux agent and sand particles, okay? There we have also recently rescued this equipment. It's a 3D scanning machine. This artifact with a laser beam will allow us to scan, in this case, the upper part of a human cranium. I think it's from Ile or Takanawan, not entirely sure. It was done some time ago. And with a software, we can get the 3D reconstruction, right? A 3D model. In this case, more recently, we scanned this mandible of a pig, and this is the model that we obtained. And we also have the printer, the 3D printer, but it's currently under repairment, so I could not show you a picture of it. And yeah, so for the equipment to take pictures, microphotographs, is an Olympus stereo microscope with a digital camera and the stream software. Okay, this version, you have to pay for this one. And for the 3D scanner, is the next engine desktop 3D scanner model 2020i, and the mesh lab software that you can get online. Okay. Okay, now some for stable isotopes that at least I wanted to introduce you to the study of stable isotopes. Again, I'm not an authority on this, but I collected some information. So stable isotopes began as a way to reconstruct ancient climate, evolving into investigating ancient human diet and mobility, and more recently entered zooarchaeology and conservation biology. So carbon-12 and carbon-13 enter plant tissues from atmospheric dioxide carbon through photosynthesis. Then moving into the tissue of herbivores and ultimately to carnivores, right? Because they eat the herbivores. The values of these carbon isotopes in bone collagen records whether the animals mainly relied on trees, shrubs, and temperate grasses, C4, tropical grasses, or camp succulent plants for their diets. Okay, so proportions of stable nitrogen isotopes, in this case, nitrogen 15 and nitrogen 14, are used to specify animal foods inputs and terrestrial versus marine foods. Oxygen isotopes, O18, extensively used in marine paleoclimate research, can also help to infer dietary intake, seasonality, and by extension, migration to habitats characterized by different O18 regimes. And I have my flirting with stable isotopes. So in this paper that we presented in 2018 in Scientific Reports, we start by, of course, getting the data from small vertebrates, small mammals in this column, and amphibians and reptiles. We get a signal, an environmental signal from the small vertebrates. This is the evolution of the small vertebrate communities plotted against time. We have all the radiocarbon data from a site called Antonina Cova, around 40,000 years of archaeological record, sorry. So once we get these signals, then we compare it with the information given by carbon-13 and nitrogen-15. So carbon-13 allow us to know the degree of canopy if the surroundings were more closed forest or more open lands, and the nitrogen contributed temperature, right, that we can compare. This is the continental signals. For the marine signals, we obtained oxygen-18 readings from the calcareous covers of zooplankton, foraminifers and ostracods, right? The oxygen-18 concentration could be measured, more negative degrees, colder environments, less negative, more warm, right? So also we can compare all these records to the polynic record, the static level, sea level, the changes in sea increases or decreases of sea, and North Atlantic signals. So all together, they can give us a very reliable, complete picture of the evolution of environment and climate in the Cantabrian region of Spain, this region. Okay, and we can, in some cases, when there are sterile layers in the archaeological site, we can complete the record with the marine record also. Okay, as for archaeogenetics, genomic analysis offers a strong set of proxies to zooarchaeologists investigating the evolutionary and biogeographical history of wild or domestic species. DNA analysis has greatly improved zooarchaeological research on actual genetic affinities of morphologically similar species. For example, ZOOM-MS, zooarchaeology by mass spectrometry. Also patterns of interspecific population replacement from past to present in various regions, and animal domestication. Research on animal domestication has benefited tremendously from the emergence of genomics and a truly global approach to the genetics of modern, economically important species and breeds. Genomic research has focused on domestic species because of their commercial uses, while wild species have been less well studied so far. Data derived from modern breed research provides insights into early domestication, and ancient DNA of the same species has refined ideas derived from surviving lineages. Finally, applied zooarchaeology aims to support conservation biologists in managing endangered or economically valuable taxa by synthesizing and conveying data on species' ancient biogeography and population dynamics. The more interest managers have in a species' long-term adaptation and ecological history, the more relevant zooarchaeological data are to them. So we can get data on bottlenecks, range shifts, translocations, extinctions, disease ecology, reconstruct ancient environments to inform management and conservation practice. Okay, and I will stop here. I want, before saying goodbye, to thank all these people that kindly let me use some pictures that they take for the DNA, for the metal and slags, pottery, mammals, etc. Thank you very much for your interest. And as the previous speakers have said, I'm totally available for questions regarding anything that I have shown today. And of course, you can contact me here in ASP or via email whenever you want. Thank you very much.